Hey everybody, welcome to the video version of evolution. We're going to cover chapter 10, genome evolution today. All right, so the teaser for this chapter introduces us to the idea that genome size, right, the number of nucleotides in the sequence of an organism's genome, varies widely across different organisms. And it's a kind of a weird trick because the differences in genome size don't actually correlate with anything you might expect, like phenotypic complexity, or really anything else we can come up with. And this is what is known as the C-value paradox. So this is something that evolutionary genomics asks. And so you can see looking at this picture here, we have the genome size of things from viruses to flowering plants. And although viruses have smaller genomes than flowering plants, why do flowering plants have larger genomes than, for example, birds? Who can say? I like to think of this chapter as being like kind of like stupid evolution tricks. These are interesting patterns that people have observed now that we have information about the entire genome of many, many, many different species. And this is mainly a chapter about people's efforts to explain these observed phenomena. Okay, so we're going to start out looking at the development of whole genome sequencing technology and talk about the evolution of genome size, genome structure, and how it differs in viruses, prokaryotes, and eukaryotes, and thinking about the selective and non-selective processes that lead to these changes. I should also point out that sometime in this lecture, I'm going to give you the access code to the quiz for this chapter. So I'll let you know when that happens. All right. So y'all are too young to live in a world when you were not aware that that people could have their entire genome sequenced. But here we are. So some of the landmark genome sequencing projects, people first sequenced a bacteriophage back in 1976, and that was not so hard because those genomes were small, right? In the 90s, we're getting slightly larger things like yeast and influenza. And then in the late 90s, like animals like C. elegans and Drosophila. And then finally in 2001, the Human Genome Project was completed. And this took 10 years. Okay, it took $33 billion. I don't even know how many people it took, but it was such an achievement at the time. I went and looked up how much it would cost to get your genome sequenced. It would cost somewhere between $100 and $200, and it would take 2 to 3 days. So we've clearly made great strides in being able to fully sequence a human genome. But the next question is, okay, now we have all of this information. What can we do with it? What can we learn with all of this stuff that we have? Okay. So one of the first paradoxes that people realized was what I mentioned before. Genomes can be enormous, right? Um, but now that we know, and now that we've sequenced the entire genome, we know that not all of those nucleotides are part of genes, right? Not all of them code for proteins. So this is a graph comparing the number of coding base pairs. So coding, meaning DNA that gets made into proteins, versus the total number of base pairs in the genome. And you can see that there is a positive relationship. So the more coding DNA you have, the larger the genome size, or conversely, I guess, the larger the genome size you have, the more of it is coding DNA. But you can see it doesn't go in a linear straight line forever. So these dashed lines are the percentage of coding DNA. And so you can see this is, if you're along this line, 100% of your DNA is coding. So like a bacteriophage or eukaryotic DNA virus, they're kind of in this range, right? Prokaryotes and unicellular eukaryotes, which would be like this brown and tan one here, are between 100% and 10%. So, they're kind of less than 50% of their genome is coded. But land plants and animals, less than 1% of our genomes are coded. So why? And why are there differences in the amount of non-coding DNA amongst groups? So a bunch of hypotheses were put forward, but I'm going to just talk about two of them. The first one was the idea, well, maybe larger genomes require larger cell nuclei and larger cells, right? Or conversely, like if you're bigger and you have bigger cells, maybe you need a larger genome. Um, and so people have asked, has there been enough selection on cell characteristics to account for these kind of enormous size differences? So we're just looking at this graph here, and here's the volume of cells. They can go from something really small. I think this is like I can't even tell what this is, which is which. Well, this one's a teleost fish, like a bony fish. So that's pretty small. And then something enormous, like a lungfish, right? Lungfish are these three green dots over here. So why is it that they have really big cells and a lot of DNA, and why is like a teleost fish, which is like not so different from a lungfish, have really little cells with not so much DNA? Sorry. And here's also a picture showing the relative sizes of different cells. So this is like a Siamese fighting fish, and also the amount of the size of the sequence. The red-spotted newt or the salamander. These have enormous genomes and also pretty big cells. Okay, but like why? Why do they need that? We don't know. All right. Second, hypothesis for why there's big differences in the amount of non-coding DNA amongst groups. Um, there could be the genome size that each of them has could just be the product of a balance between a couple different things that are happening. One is the spread of self-replicating genetic units, so transposable elements or transposons. Um, might be trying to make the genome bigger and bigger and bigger and bigger. But then on the other side, there could be selection for efficiency. So, trying to replicate your DNA quickly, and not wasting a lot of ATP in the process of managing all that DNA. So you could have a push and pull between things that are trying to make the DNA bigger and things that are trying to make the DNA smaller. Um, and the solution that different taxa may come to may be different because different taxa have kind of different evolutionary history and different environmental challenges. And so maybe the optimal solution for the newt is not the same as the optimal solution for the lungfish. Um, Other elements are that when we're talking about unicellular organisms, they have huge population sizes, right? So even if there's only a small effect of selection, a tiny little selection differential, because of the huge population sizes, it is likely to have a larger effect in eliminating even slightly bad non-coding DNA like transposons. If you have a fairly small population size, um, selection, things are going to have to be, I don't know, selection is going to have much more variation in fitness to be able to eliminate things. So perhaps just like the tiny, tiny little bad effect of a transposon might not be enough for selection to act on it if you have a really small population size like you would in an animal or something like that. I say small, but I mean like small compared to like a bacterium. All right, so I didn't answer the question because we don't know the answer to the question, but we will continue. Um, okay, so people have asked, well, there's a little bit of a pattern for things that have more protein coding genes to be more complex, but not the pattern doesn't hold out if you add more and more animals and plants and everything else to it. So just overall, the number of protein coding genes or the G value doesn't scale with organismal complexity. So the blue represents the number of coding genes and the orange represents the protein coding sequence. Um, so yeah, I don't know, like a slime mold, right? Not that complex. Um, so this is the blue for the slime mold. Fruit fly, is that not more complex than a slime mold? Who can say? But it certainly has fewer coding genes. We can look across. So for example, Coelicans, a nematode, has actually more coding genes than we do. A pufferfish has more coding genes than we do or a mouse does. There's not really a rhyme or reason to what we're seeing here. All right, but what are our hypotheses? One thought is that the network by which genes are regulating the expression, the the network of regulatory genes that are kind of controlling the expression of coding genes might be affecting phenotypic complexity more than the actual number of protein coding genes. So it could be all the stuff that's not getting made into proteins, but is instead um, influencing how protein coding genes are working. That is more important. And so if you look like nematodes, fruit flies, and humans all have similar numbers of protein coding genes, but what's not indicated by this picture here is that they differ in the number of transcription factors that they have. They also have big differences in regulatory complexity. So it is possible that it is regulatory complexity that scales with organismal complexity and not the number of protein coding genes. The second thing that might explain the G-value paradox is that all these non-coding regions may also be regulating genes. I always ask people if you remember microRNAs, and then here I'll ask my imaginary class, does anyone remember microRNAs?
I have no idea what you're talking about.
Okay. What about you in the corner over there, old timer? You sat through all of my lectures for 1114?
Oh yeah, I totally remember microRNAs. Those are those little pieces of RNA that interfere with transcription.
Um, yeah, well, actually, yeah, you got it. They get in there and they stick to the DNA and they interfere with transcription. So if they interfere just right, they can regulate how genes are expressed. All right, that's the first thing that is in the non-coding region that may be regulatory. And the second one is that some transposons, even though they do not copy themselves to do anything useful for us, actually have become changed now and end up serving regulatory functions. And the third possibility here, and again, these are not necessarily mutually exclusive, but these are all potential answers for why the protein coding genes, number of protein coding genes don't scale with organismal complexity. Um, the third one is that alternative splicing. Anyone remember what that is? Anyone? Anyone?
All right, I'll explain. Alternative splicing is just like when you take out different introns.
Okay, yeah, you take out different introns. So why does that matter?
It makes a different protein. Jeez.
Okay, okay, don't get salty about it. All right. Yes. So if you have alternative splicing, more than one gene with can be or one gene can produce multiple different protein products. So you can get complexity in other ways as well. All right, let's take a look at the genomes of different kinds of organisms um, that we have want to take note of. So viral genomes are especially interesting to us because they are incredibly diverse. They can, virus can have either DNA or RNA as its genetic particle. Some of them are retroviruses. Some of them have DNA that's single-stranded. Some have double-stranded DNA. Um, they are either positive or negative sense, meaning that they use different strands for coding. Some are circular, some are segmented, some of them are haploid, some of them are diploid. So a huge amount of diversity. Later on in the course, we're going to talk about RNA world. So there is some evidence that viruses preceded cellular life. And one of the pieces of evidence of this is that they have some genes that other kinds of cellular life do not have. Um, so if we think about it this way, the kind of viruses that we have amongst us right now might be from the original virus world, and they may be the only remaining survivors in this world full of things that have cells. Okay, one thing that all viruses share is that their genomes are quite compact. So this is a picture of, forgot who this is right here. Let's see. Can anyone help me remember which virus kind of looks kind of influenza-like, doesn't it? I'm looking through my textbook because I can't access any of my notes while I'm in Zoom. All right, what do y'all think it is? Anyone?
I think it's influenza.
Why do you think that?
Because of the spike proteins.
Um, Not quite. This is SARS-CoV-2. All right. And yeah, and this is just showing the very compact um, little squiggle um, of RNA that it has inside of it. Here we go. Here's our corona and our influenza B genome. Well, actually, the other one did have a corona, so you were right.
See?
All right, good. Here's a picture of the influenza B virus. It is single-stranded, negative sense RNA virus. And unlike the coronavirus, it doesn't have a single chromosome. It has 8 different chromosomes, and they're not circular. They're instead linear. And they encode 11 protein coding genes. And so these are the 11 protein coding genes we can see here. And one interesting thing about the influenza genome is that some of the genes partially overlap with other genes. And so they can be, you can get different protein products depending on how they're read. Um, and so you can see right here, you can actually just see that pretty much every single thing that you need to be an influenza particle is contained right here. So each of the segments, the color goes with the color of what it makes. So you can see how every single thing here codes for all of the parts of the influenza particle. All right. Here's a think pair share. Would you expect to see greater linkage disequilibrium between the HA over here and the NA locus of influenza virus, or between the S and C loci of the hepatitis B virus? Explain.
Are you gonna make us work in groups?
I mean, not if you don't want to. You could work individually.
Oh no, I want to work in groups. Come get me.
All right. Let's not do that right now. Okay, so do you remember what linkage disequilibrium is?
Oh yeah, I totally remember.
All right, what is it?
Uh, uh, uh. Oh, I know what it is.
Oh, I didn't see you over there. I'm glad you showed up.
Yeah, am I right? Linkage disequilibrium is like when you unexpectedly find linkage between two loci, so that if you see one, you're more likely to see the other. So it's like you're not getting recombination.
All right, that is a pretty good description. All right, so where do you expect to see less recombination in hepatitis B or influenza?
Well, well, like it looks like hepatitis B. All the DNA's all stuck together in a big clump.
Yeah, it is just a single circle.
So, like, how the heck are you supposed to have recombination when you just got one circle?
Yeah, that's right. You don't expect to see recombination when you just got one big circle.
All right, then. That's the answer.
All right. So yeah, you would expect to see more recombination in influenza between the HA and the NA than you would in hepatitis B between the C and the S. All right, good job, everyone. All right, the next question that people have asked is, why is it that viruses are able to have such tiny, tiny little genomes? And here's just another example here. So this is the hepatitis B virus. And this is a virus that, well, it's very small. It's a DNA-based virus. It's circular and double-stranded. So, we have double-stranded DNA, but we have, a big genome. So how does hepatitis B manage this? Um, I mean, we can understand why it would want to be small, just because the viruses that can replicate more quickly have more generations, more opportunity for mutation, more opportunity for spread, right? So there's a lot of natural selection for rapid replication. And viruses have come up with cool tricks to be able to have small DNA so that they can replicate quickly. And one of their cool tricks is they have these multiple partially overlapping genes that are in the same reading frame, and then also multiple genes in different reading frames. Um, so do you remember what is a codon?
Yeah, codon it's totally like a set of three nucleotides that are rent together to make a protein.
The whole protein?
Just an amino acid. Ah,
All right, good. All right, so the ribosomal RNA, do you remember? It's translating that messenger RNA into proteins, and it's doing it three nucleotides at a time, and the transfer RNA is bringing over those amino acids to add to the chain, right? Okay. So it could read it this way. GNU are two new hot ads. Okay, whatever. So this could be a single product. You could also arrange to have transcription, then translation start here, right? And then that will give you a different product, and then here will give you a different product. So this is the same reading frame, but if there are different start and stop positions, you can get different protein products. Alternatively, if you have different start and stop positions here, you can change the reading frame and also get different protein products. And so this is a map of all of the products that are made out of this single, oh, I'm sorry, well, double-stranded, but it's a one double-stranded loop here. All right. So mobile genetic elements like plasmids, prophages, and transposons can influence the organization and content of the genomes of prokaryotes like bacteria and archaea. And they have the capacity to both move within and between genomes. So we're going to talk about some of those here. Here's an example of the E. coli genome, one of the strains of E. coli. And it is obviously it's a circular chromosome. Um, but within the E. coli, it not only has this big old circular chromosome, it has a couple of really important plasmids. So it has a specific plasmid called the virulence plasmid. So if it has this, it becomes a virulent strain of E. coli. Without it, not virulent. Does anyone remember what virulent means?
sure. It means it's really bad for your health.
Yeah, that's what virulent means. They also have mini plasmids that do other things as well. One other interesting feature about these genomes is they have what are known as genome islands. And so these are in yellow. So if we look at the lineage of E. coli, we can see that these are small little clusters of genes that have been horizontally acquired. So they've been acquired in a lump from somewhere else. And so some of those do things like, um, you know, resist tellurite, transport iron, you know, there's a phage colonization factor. They do all sorts of things. Okay. Um, but we'll return to transposons and everything else later. Okay, so comparing the genome size of prokaryotes to the number of protein coding genes, right? We talked about this a while ago, but we just kind of looked at all prokaryotes lumped together. Um, so the prokaryotic genomes are larger than viral genomes, okay, but they are still quite compact. So moving on from viruses to bacteria and archaea, they are 85% to 95% protein coding. So they, um, you know, are not as efficient as viral um, genomes, but still pretty good. Some things they do not have, they don't have spliceosome introns, so they don't. Remember what's an intron?
Yeah, that's the piece of RNA that gets like cut out.
Yeah. So instead of having spliceosome introns, they have these self-splicing introns in the ribosomal RNA and the transfer RNA. So they kind of splice a little differently, um, not the way that we do. And they also have fewer pseudogenes and transposons. Um, all right, so we'll talk more about that in a minute. What is a bacteriophage? Anyone? Anyone?
Oh yeah, bacteriophage is like a parasite.
Okay. Can you get attacked by a bacteriophage?
Nah, dog. You can't get attacked by a bacteriophage if you're a person. They only attack bacteria.
Yeah, that's true. All right. Does anyone remember what a prophage is?
It's probably not important.
I mean, it's kind of important.
I don't remember what it is.
All right, let me show you what a prophage is. A prophage, do you remember that viruses will put their DNA into the DNA of the bacterial host, and then that DNA will, by crossing over, end up incorporated into the host DNA, and so then the host is stuck with a little piece of the bacteriophage DNA. Do you remember that?
Oh, yeah. That's the little piece that makes more virus particles.
Yeah, so that little piece of DNA gets made into the virus particles, and that's what allows the virus to be able to spread. Okay. So these little inserted prophages, or little bits of the genome of the bacteriophage, can remain latent for generations. So they just sit there in the genome, and the bacteria is going about its business, it's copying itself, and the prophage just gets copied along with it. And so prophage DNA can make up to 10% of the genome of a prokaryote like a bacteria. And um, so sometimes that prophage DNA can acquire mutations. And so if the prophage DNA gets a mutation in it, then it doesn't work as a prophage anymore. The virus can't replicate using that little bit of DNA. And so you keep getting more prophages being added, and some of them are being, just disabled accidentally by mutations. And this is one way in which you get differences between species or strains of bacteria that we can easily detect. So we can see here's three strains of Streptococcus pyogenes, and we can identify the M1 from the M19 from the M3 based on the number of prophages they've accumulated. Some of these prophages can encode virulence factors. So, bacteriophages will often have the genes that allow bacteria to to basically be virulent. So, they can kind of give the bacteria some tricks that allow them to be able to evade their eukaryotic host's defenses. And so, this is another way in which the bacteriophage DNA that gets incorporated into bacteria is important. All right, what's a plasmid?
Oh, it's that little loop-de-loop.
Okay, what a loop-de-loop of what?
Like genetic material.
Yeah. Where is it?
It's in the nucleus.
Yeah. Do we have them?
No,
All right, who has those?
like bacteria, archaea.
Yeah. So plasmids, they're not the main big circle of essential DNA that's in the bacteria. The plasmids are non-essential. They're circular. There could be one, there could be many copies. Some of them are small, some are big, but some of them can be really useful for their bacterial hosts in particular ecological circumstances. So we have found these little plasmids that may code for things like resistance to antibiotics or resistance to heavy metals. We also use these a lot in the lab because you can easily give plasmids to a bacteria to make it resistant to antibiotics, or you can manipulate a bacteria to be resistant to heavy metals so it can be used for bioremediation. All right, let's talk about other ways in which you get genetic material into prokaryotic genomes. So the general phenomenon of moving stuff into the prokaryotic genome is called horizontal gene transfer, okay, which we abbreviate as HGT. Weirdly enough, it's also called lateral gene transfer, which doesn't really make sense because, like, you think horizontal, I guess horizontal and lateral, do they mean the same thing? I'm not sure, but both of these are used interchangeably. And when bacteria and archaea are able to get genetic material like this, it allows them to gain genetic variation. Um, so there's three main modes of genetic recombination by horizontal gene transfer. The first one is transduction. And so this is a picture of a bacterium, and a bacteriophage is landed on it, and it has injected its own DNA into the bacterial host. The phage DNA just kind of squishes around in there, and then by crossing over, it ends up, um, excuse me, incorporated into the host bacteria's DNA, and then that host will, kind of copy it up. Okay. But sometimes in the process of all that scrambling around, some of the host's DNA gets packaged into the viral capsids. And so when the bacteriophage kind of explodes the bacteria and all the capsids go out there to attack another bacterium, it ends up being not just the virus or the phages DNA that ends up in the new bacterium. It could be just a little bit of the previous host's DNA gets transferred. So this is a way in which genetic material can get passed from a bacterium to a bacterium. Okay. It's not that the bacterium wants to do this. It doesn't really have any say in it, but it is a mechanism that does introduce genetic variation. The second process of horizontal gene transfer is transformation. And so this is like I've described it before, like how bacteria are like those guys on the like the furniture fixer upper, channel where people are going down and picking up furniture off the curb and like cleaning it up and selling it again. Like bacteria are out there just picking up whatever pieces of free DNA fragments they can find, and they're like, I can do something with this, and they take it up and they incorporate it into their own genome. Okay. So, and yeah, the incorporation happens by recombination. So first the little squiggles of DNA are just taken up within the cell, and then through a process similar to crossing over, those little squiggles of DNA from outside sources end up getting incorporated into the host DNA. We referred to pathogens that can take up these pieces of DNA as being competent. Um, and yeah, when they take up this DNA, they're using it for some various sorts of things. Sometimes they'll use it for quote unquote food. They're not actually eating it, but they're just like breaking up the particles, the molecules, and recycling those molecules to make its own DNA. Or, using it to, for example, repair a patch of its own genome that's been damaged. But the upshot is that it helps the bacterium generate variability. All right, the third kind of genetic recombination by horizontal gene transfer is conjugation. And so this is when two bacterium line up, they form this conjugative pilus, and they exchange genetic material. Excuse me. Sometimes it's like a plasmid that goes through the pilus. Sometimes it's part of the main chromosome that goes through the pilus. All these things are possible. All right, y'all. Which of these three processes, transduction, transformation, or conjugation, is least likely to have evolved by natural selection for its present purpose? Okay, so which of these processes is least likely to be an adaptation?
That's a tough one. I don't think it's a tough one. Why not? Well, like, the bacteriophage is just like a parasite. I think transduction couldn't have evolved as an adaptation. It's just like a thing that happens. Well, I guess so.
All right, I think you're right about that. Conjugation clearly evolved for, the function of exchanging genetic material. Transformation, um, it definitely helps the bacteria pick up the raw materials to make more DNA, and to repair its own DNA. So, sort of doing what it's supposed to be doing. But transduction is least likely to evolve for the purpose of exchanging DNA. It just happens to happen that way sometimes. One thing to note is that horizontal gene transfer is not the same thing as sexual reproduction. So it's not like sexual reproduction for prokaryotes. Some differences. Well, horizontal gene transfer is not limited by species or even the domain. Um, and so you can exchange materials between like archaea and bacteria, between archaea and eukarya, in addition to just between different species of bacteria. There are costs to horizontal gene transfer, so um, the transferred genetic material might have been super beneficial in the species that it came from, but it might actually reduce fitness in the bacterium that it receives it, especially if the environment is very different than the donor species environment. Some benefits of horizontal gene transfer. Sometimes, just by, by good luck, those little transfer genes increase the fitness of the prokaryote who gets them. So some examples of that, there are these little sections of genetic material called pathogenicity islands. Um, and these are horizontally transferred stretches of DNA that have the genes in them that allow a bacterium to basically go from a commensal to a pathogenic lifestyle. So anyone remember from 1114, what's commensalism? Commensalism, anyone?
Yeah, I'll do that one. So, like, commensalism is like when you have like two members of an interaction and one of them's benefiting and the other one is like doesn't care.
Yeah, that's it. So, for example, we have a lot of E. coli in our gut, and some of it's beneficial, some of it is commensal, but then some of that could potentially become pathogenic. So, just having those right genes could turn a bacterium that's just minding its own business somewhere into a pathogen. And most importantly to us, for this course, evolution, horizontal gene transfer supplies variation for natural selection. All right, I'm going to shift topics a little bit to just think a little bit about how we can identify similarities or differences in prokaryotes in gene order. And so by gene order, I literally mean like if you look along the bacterial chromosome and see like what codes for what, like as shown by this color here, how does that line up with how other bacteria code for things? So this kind of a graph is called a syntenic dot plot, and it's comparing the order of genes in two different strains or species. So you can, for example, see translocations, inversions, deletions.
I don't remember what those are. Ah, do I have to do everything? Fine. A translocation is like when you get a piece of DNA that ends up getting moved from like one chromosome to another, or like an inversion's when it goes backwards, deletion is missing.
All right, thank you. You're very good with those kind of chaotic things, aren't you?
Yeah, that's my specialty.
All right. So we can use these kinds of syntenic dot plots to look at these kind of patterns. So if we imagine here, this original bacterial chromosome goes here from purple to green, back to red again, and it had an inversion that happened here. So you can see this little section got flipped around. Okay. Um, so if we compare the genome of this guy over here, number one, to number two, we're going to see that mostly so if everything is the same, it should follow along this axis here. So you can see the red is the same, the yellow is the same, and up here the purple is the same. But when we get to that section in the middle, we can see it's going in the opposite direction, indicating the inversion. All right, so let's compare this one over here to this other species here. So this one, it still has the inversion from number two, and it has a second inversion over here. And so we can see the second inversion here and here. So the main diagonal that I showed you before means that they are the same, and then the off diagonal is showing that they're different. All right, how do you interpret these two syntenic dot diagrams? Somebody want to look at that first one there? What's going on? So we're comparing E. coli K12 strain B to E. coli K12 strain A. So what's going on with the green line?
Oh, they're the same.
Yeah. And what's that red line there?
It's an inversion.
Yeah. What do you think all these little smattering of dots are?
I don't know, just like different mutations that make them completely different.
Yeah. What's going on in this one here?
That's chaos.
It does look like chaos. So does it seem like those two strains, the Syringium and the Myxococcus, have similarities in gene order?
No way, man.
All right. All right, that's what we got here. So, yep, in this one, you can see that for the first plot, most of the genes are in the same place in both, and in the red line, there's a chromosomal inversion along one of the two lineages, which is doing that. And then in this species, you find that all the similarity in gene order has disappeared altogether. All right, another weird pattern that people have observed looking at genomes involves codon usage bias. So, all right, so we all know that, for example, you can make leucine in a whole bunch of different ways. So all of these codons code for leucine, right? But for whatever reason, CTG is the one that's most commonly used to make leucine as opposed to any of these other ones. Um, The first thing we know is that transitions are more common than transversions. What does that mean?
Oh, oh, wait, I know.
Yeah, what is it?
Like, if you have a purine, it's more likely to become a purine, and if you have a pyrimidine, it's more likely to become a pyrimidine.
Yeah, that's it. So purine to purine is transition, purine to pyrimidine is a transversion. Um, and another weird thing is that the frequencies of the transfer RNA molecules that are bringing the amino acids to the growing amino acid chain, those are not equal. Um, And so you find, what the picture showing here, that a more common codon is translated more quickly with less error. Oh, I think this picture just came up a little late. It's just showing the relative frequency of transitions to transversions. Yeah. Another weird pattern. So, we've talked about like microsatellites, right? Mini satellites, mostly microsatellites. And so that's what, like repeated sequences of like 3 to 5 nucleotides. Um, right? So you get like ATC, 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 like that, right? But you don't get longer repeats. Okay. And those are significantly underrepresented. So here you get a lot of like 1, 2, 3, 4, 5, 6, well, five, nucleotides in a row of repeats, but then beyond that, you get less and less. So you don't really ever get things that are more than 9 or 10 nucleotides repeated in E. coli. A little bit more likely to get it in larger things like yeast or nematodes, but yeah, but you can still see that there's this pattern here that you don't get long repeats. And this gives us the thought is that there may be evidence of selection against inaccuracy in replication, transcription, or translation that are caused by these repeated chunks. Just a picture. What does it mean? Ah, hey guys, what's an intron?
I said that one. That's an intervening sequence that gets cut out of the RNA to make messenger RNA.
Yeah. What's an exon?
That's the express bit that gets made into proteins.
Yeah. All right. Does mRNA processing affect transcription or translation?
I forgot what mRNA processing is.
Oh, well, y'all, do you remember that there's 3 things that you have to do to make RNA into messenger RNA? Can you think of what any of them are?
Oh yeah,
Oh, who's that?
I'm over here.
Oh, okay. What name some of the things?
You gotta put a cap on one end, you gotta put tail on the other end, you gotta cut out all them introns.
Yeah. All right. So when that happens, does that affect transcription or does that affect translation?
Oh, it's gonna affect translation because it happened after.
Yeah. All right. So Ackerman and Chow asked whether there's selection against inaccuracy in transcription or inaccuracy in translation. So what they did is compared repeats that spanned introns to repeats that spanned exons. Um, so if you have a repeat, so here's a whole bunch of A's, and it goes like through one side of the intron and not the other. Okay. So if you have a repeat that goes across the intron, um, it's not going to reduce the accuracy of replication or transcription, right? But it might affect the accuracy of translation because translation is the thing that happens next. Okay. And so what they found is that repeats spanning introns are more common than repeats spanning exons. So accuracy in translation has less of an impact on fitness than accuracy of replication or transcription. So that is an interesting observation. All right, one other weird trick that people have figured out, which you may have read about before, is that organisms vary in their ratio of adenine and thymine to guanine and cytosine. So for example, but in weird ways, right? So here's streptomyces. They got more GCs and less ATs. But what about Plasmodium? Oh, they have less GCs and more ATs. Why? So there's like different biases in different species. Like for us, we got more ATs than CGs. Um, but why though? One thing we know is that transitions from guanine to adenine and from cytosine to thymine are common, but transitions from adenine to guanine and thymine to cytosine are less common. Like this is the kind of thing that people just figured out by just looking at stuff and they just found that out. So that's the observation. So there appears to be mutation that just the way in which mutations happen seems to be driving genomes towards less G and C content, right? But you can see over here, these guys have more G and C, right? So, so explain that, right? So the mutational bias towards less GC content can't explain why some species have more GC than AT. So something other than the kind of natural way that the mutations happen must be elevating GC content. One thing that people have found that it can influence GC content has to do with gene conversion. So let's talk about what that means. Okay, so here you have two homologous chromosomes. What's it mean that they're homologous?
It's a matching pair.
Yeah, it's a matching pair. So these are the two strands of each of the homologous chromosomes. And the red one has a double-stranded break.
Ouch.
Yeah, right there. Okay, so the homologous or the blue is being used as a template to repair the break, right? So the repair mechanisms are reading the sequence of the blue to put in the gap for the double-stranded break. Um, and so due to the biochemistry of the repair mechanism, if you have a strand with a G or C, it's more likely to be used as a template than a strand with an A or T. I can't exactly tell you what the biochemistry is, but I'm sure you can think of somebody who you can ask who probably knows the answer. Okay, but because of this mechanism, the adenine thymine pairs can end up getting converted to GC pairs when the repair is happening. And so that could potentially like soup up the number of GC pairs. All right. All right, so that maybe answers that question, but then other kinds of questions people are asking. For example, looking at something like E. coli, right? What fraction of that E. coli was acquired by horizontal gene transfer, and when did that happen? And so they scanned the genome of this strain of E. coli for areas where the base pairs or the codon usage was different than the rest of the genome. And from doing this, they found that approximately a sixth of the E. coli genome came from horizontal gene transferred. And a lot of those genes had only been transferred quite recently. All right, so let's review a little bit. Tell me how DNA is replicated, okay? Make sure you include these terms in your description.
I love DNA replication.
Okay, are you going to tell us about it?
Okay. All right, so let's see. The two strands are separated from each other, and then DNA polymerase comes in and it adds like nucleotides to the one of the strands, but the other strand is going the wrong direction. So like the leading strand, the DNA polymerase adds stuff in order, but then the other strand is kind of backwards, so DNA polymerase has to kind of fly in and stick the nucleotides in little pieces.
Yeah, that's pretty good. Okay. Be more helpful to have a picture, but that was very good for no picture. All right, so in eukaryotes, where does transcription take place?
Transcription
Yeah, where does transcription take place?
in my office,
No, not in your office. Where else does transcription take place?
in the nucleus.
Yeah, in the nucleus. All right, where does translation take place?
Outside of the nucleus?
Yeah, outside of the nucleus, in the cytoplasm. All right, so for prokaryotes, where does transcription take place? Anyone? Anyone?
I think it all just takes place in the nucleus, like the whole thing, everything.
All right, let's take a look. All right, so in prokaryotes, everything is kind of happening in the nucleus here, and there is a bias where the important genes tend to be on the leading strand rather than the lagging strand. So in prokaryotes, instead of this being like a stick of DNA, it's a loop of DNA, and this is the origin of replication. So this is where the two strands are separated from one another, and then DNA polymerase comes in and adds nucleotides. So this is the leading strand. So it's oops, no, it's not. This is the lagging strand. Where's the leading strand? Oh, yeah, this is the leading strand. So the DNA polymerase is just adding, going this way. And this is a lagging strand. Um, and so as a result, it is able to add a little bit. Um, and then the bubble opens a little more, and then it jumps back and adds more, and then it adds a little more, and then it adds a little more. All right. So during transcription on the leading strand of the chromosome, the RNA polymerase is moving its way away from the replication fork.
Wait, what's the replication fork?
It's just a name for the place where the bubble is forming. It's like the little corner of the bubble as the two strands are being pulled apart.
Oh,
All right, so the RNA polymerase is copying or is making a strand of RNA as it's moving its way from the replication fork. And because it's doing it this way, it makes it less likely that RNA polymerase and DNA polymerase are going to have a crash, or that RNA and RNA polymerase are going to have a crash. Um, Meanwhile, on the lagging strand here, the RNA polymerase is moving towards the replication fork. So you can see it's going towards the replication fork. Whoops. um, which increases the probability of a head-on collision. Um, so this is one reason why you might want to have the important stuff on your leading strand and not on your lagging strand. Because if you have a head-on collision between the RNA polymerase and the DNA polymerase, could potentially cause the wrong nucleotide to be brought to the strand. Consequently, you end up with more guanines on the leading strand than the lagging strand, which causes this kind of a skew where you have, I guess the GC skew is referred to as guanines minus cytosines over the total guanines and cytosines. If you have guanines and cytosines on this at the same frequency on each strand, then the skew would be zero. So this is a picture of Clostridium, a Clostridium bacteria, and you can see that the genes, let's see, so what do we got here? They're predominantly on the leading strand rather than the lagging strand. So this green part here is the leading strand, and then the inside part here is the lagging strand. And then here's the leading strand, and then, yeah, okay. And we can see the bars are the places where the coding DNA is. So you can see on the inner orange ring, this is just showing like the relative proportion of GC skew with an excess of G over C on the leading strand. All right. Now let's move from prokaryotes to eukaryotes. So looking at their nuclear genomes, yeah, way back a long time ago, before the first human genome was sequenced, people assumed that if they just sequenced the whole thing, they'll just find more and more and more genes. But they were shocked to find that there are only 1.5% of the human genome that is made up of coding genes, which is pretty typical for eukaryotes. Um, yeah, so it's about a quarter introns as well, a quarter transposable elements, including um, lines and signs. I cannot remember. They stand for L stands for long, S stands for short. but I can't remember the rest of the abbreviation. Other components that are non-coding include like heterochromatin, which is involved in the winding, segmental duplications that are caused by gene duplication events. So, sometimes you get like gene copies that aren't used anymore, microsatellites, which we've talked about before. Okay. Um, so um, yeah. The most common transposable element that you see in humans are called line 1 or L1 elements. And these are referred to as autonomous transposons. So the autonomous part means that they actually make the enzymes necessary to catalyze their own movement elsewhere. And so we see a number of cases in which a line 1 element um, transposed itself somewhere else in the genome, but then once it got there, it had some kind of a mutation that made it unable to move. So we can see lots of copies of these L1 elements in our DNA. Let's see. So sign elements are non-autonomous transposons, and so they require an enzyme called transposase from another transposable element to allow them to be catalyzed to move. So we can see here, looking at the history of primates, that we have acquired a bunch of very specific line one elements called ALUs. And so we can kind of look. So there's, more than 850,000 ALU copies within the primates. Humans alone have 495 that our nearest ancestors did not. Yeah, so ALU elements are signs. Oh, I'm sorry. I said they were lines. I guess they're signs. They're short. And yeah, so we can see lots of evidence of different kinds of ALU insertions. So what is a transposon? It is a selfish genetic element. It is a little piece of DNA that is just trying to increase its own fitness without regard to the entire organism that it's in. So all it's trying to do is replicate itself and put it in the genome more times. When it does that, it is in no way benefiting the whole organism. It is only benefiting itself. So it creates an extra transposon copies to prevent it from getting deleted, right? So it's spreading itself throughout the organism and preventing copying. So you get a transposon, it'll copy itself into a new site on the chromosome, and then potentially the host deletes it, and the new one is still there. Um, You can get transposition onto a plasmid that moves a transposable element into a new genome entirely. So the transposon could end up in a plasmid, and then that could end up, through conjugation, ending up into a completely new chromosome. If you have something diploid, like us, then the transposition can copy an element from one chromosome to its homologue. So here you have your homologous pair of chromosomes, and it starts out here, but it is able to, so okay, put it this way. If it's only in the one side, then only, remember that we're diploid, and we will put one of our two chromosomes in each of our sperm or eggs, right? So if it's on only one of the two homologous chromosomes in the pair, then it has a 50-50 chance of ending up in a new human being, right? However, if it's able to copy itself into the other homologous chromosome of its pair, then it has 100% chance of no matter which chromosome ends up in the offspring, they will all have this transposon. All right, so if a line 1 transposon is viewed as a selfish genetic element, why might we view ALU elements as hyperselfish genetic elements? I'll give you a hint here. Line 1 are autonomous and ALU are non-autonomous.
Oh, oh.
All right, do you need a hint? What can you do if you're autonomous versus non-autonomous?
Well, if you're autonomous, then you got your own enzymes, you can copy yourself somewhere. If you're non-autonomous, you have to sit around like a slug waiting for someone else to make you an enzyme.
Yeah. So why might that make you hyperselfish?
Oh, because then you're both like a parasite on the host and a parasite on the other transposons.
Yeah. All right, so transposons can have fitness effects. Um, So depending on where they insert, they can end up like they can insert into the middle of a gene, which can totally wreck the protein it's trying to make. They can insert into a promoter and or near a promoter, and then it causes, the gene expression to be messed up. They can um, drive inversions or deletions or translocations or rearrangements. They can create RNA interference, um, or yeah, the host can enact RNA interference as a defensive mechanism. So the host can put in a little piece of microRNA to be able to prevent transcription to avoid the transposon, and that could affect what the phenotype is. Occasionally, a transposon can be beneficial. Um, so we can see here an example of 2 Arabidopsis, which is a plant, and they differ by an inversion and a deletion. Okay. And we can also see the genome structure has been really reshuffled after the divergence of mouse and human lineages in a way that the transposons have contributed to. All right. Um, all right. At this point, I'm going to tell you what the access code is for the quiz, but you should still listen to the rest of the lecture. Okay? The access code is capital G dash whiz, W-H-I-Z. I'll say that again, capital G dash W-H-I-Z. Gee whiz. All right. Um, let's start this part by just reviewing some terms. So what's a centromere?
Oh, it's the dot in the middle of the chromosome.
Yeah, it's a little sticky spot that holds the 2 sticks of chromosome together. Um, what is a polar body?
Oh, isn't that like when you make a like a an egg and then you don't make 4 eggs, you make 1 egg and 3 little blobs.
Yeah, those blobs are the polar body. What's non-disjunction? Oh yeah, I know what non-disjunction is. That's like uh that's like when you have linkage between 2 loci. Yeah, between 2 loci. Right. All right. So, where do you get centromeres from? Like, why do we even have these things, right? Like, not all kinds of chromosomes have centromeres. One hypothesis is called the centromere drive hypothesis, which is that selection favors mutations to centromeres that increase their chance of segregating to the oocyte instead of polar bodies. All right, so what does that mean exactly? Just remember that you start out with 2 sets of chromosomes, right? And in meiosis, there are 2 sets of uh, divisions, right? There's meiosis 1 and meiosis 2. And so the process of meiosis 1, so first you double each of your chromosomes, then you have like 4 copies of each thing, not copies, but 4 chromosomes, two pairs, and then you divide them in half, and you divide them in half and again. So by the time you get to the end, you have 4 cells that have 1 stick chromosomes in each of them, right? But in females, what you end up with is you end up with 4 cells that each have like 1 stick chromosomes in each of them, but only one of them gets all of the cytoplasm and becomes the ovum. The other ones become polar bodies, and um, so one hypothesis is that mutations are the result of, centromeres are the result of mutations that um, that are kind of causing the chromosomes to try to get picked to be the one to go into the ovum instead of ending up as a polar body. And so if you have a centromere, it might increase the number of repeat sections for the microtubules to bind to.
Wait, what's a microtubule?
Oh, you know what a microtubule is. What do you think it is?
Oh, wait a minute. Is those grabbers in meiosis that like grab the middle of the chromosome and pull it apart?
Yeah. So if you have a big sticky part in the middle, it might be just kind of you know, um, like a grappling hook that's being used to grab onto the microtubule to make it so it's more likely to end up in the ovum. Centromere drive can actually cause problems. It can cause non-disjunction.
Oh, wait a minute.
What?
I think I we said what the other thing was wrong.
Oh, wait, did I read non-disjunction as Yeah, it's kind of late. Yeah, whatever. Okay, non-disjunction. Non-disjunction is referring to the fact that sometimes you get chromosomes that are being not pulled equally in meiosis.
Do I hear a dog?
Let's just ignore that dog. Yes, sorry. Non-disjunction is when you have sets of chromosomes and they get pulled apart unequally. Um, so, um, okay, so you start out with your, kind of four sticks of chromosome here, two copies of the one from your mom, two copies of the one from your dad. They're all stuck together in prophase, getting ready to be pulled apart, right? If you have an especially sticky centromere, it can cause the chromosomes to not separate correctly. And as a result, you could end up with double the number of chromosomes in one cell that you're supposed to. And so one thought, so yeah, so there is some evidence of that looking at SEN-H3 histone mutations, which we think may be there because they resolve the problem of the centromeres just trying to get stickier and stickier and stickier. Um, because obviously if they are too sticky and they cause non-disjunction, the organism that the chromosomes are going into is probably going to die, right? So there's been selection against getting too sticky. Um, yeah, so Heinekoff was able to deduce that by rapidly generating genetic differences in meiotic machinery like SEN-H3, et cetera, of closely related species, um, you could potentially end up by centromere drive having reproductive isolation and speciation. So differences in the way the centromeres behave could make it so that you either can't have species interbreeding with each other, but also if you have a small mutation like this, it might make it so that that within that population, some individuals can't reproduce with other individuals. All right, what's a telomere?
That's the egglet at the end of your chromosome shoelace.
Yes, it's the end of your chromosome, like the aglet on a shoelace. What's telomerase?
It's an enzyme.
Well, we know it's an enzyme because it ends in ase. What do you think it makes?
Telomeres.
Yeah, I think that's a good guess. So telomeres are those long stretches of these short repeats that end some of the eukaryotic chromosomes. And by end, I mean like they're literally at the end of the chromosome. They're on the little roundy tips. Um, and they may have arisen as a solution to a problem that happens when you have linear chromosomes instead of circular chromosomes, which is that when you're replicating and replicating, you end up with these little incomplete areas that may not be easy to copy. Um, So telomerase is an enzyme that has an RNA template, and it just adds a little repeat section of the 3' end to the end of the chromosome, and it prevents you from losing the coding sequence, by the chromosomes just getting shorter and shorter and shorter. All right, another question. Why do we even have introns, right? Um, so if they're not getting translated, why are they even there? This is explained by the exon theory of eukaryotic genes. So, um, just the idea of genes being introns and exons is very, very ancient. And many of our current genes happened because exons got rearranged. And so, if we didn't have these introns, then we would end up with this unequal recombination. So, when you would have crossing over and exchanging pieces of material, um, the protein domains might get kind of messed up by having unequal lengths of genes crossing over with each other. So the thought is that just like having these little introns gives you a place to have the crossing over. Um, when you have introns, you have longer genes, right? And so that separates the exons and gives you more of a chance of recombination. Can you picture what I'm talking about?
Yeah, you got your like chromosomes and like the exons are like the loci you're thinking of separating by recombination and like then now they're farther apart so you can have more recombination.
Yeah, that's it. So you get more recombination between exons possible, and this gives you the option for having new proteins that you did not have before. All right. Um, what's linkage disequilibrium?
I think that's what I said last time by accident.
Yeah, linkage disequilibrium is when you have loci that do not get recombined by random. Instead, they are linked to each other. All right, so the rate of recombination varies across species. So there are some trends. So you can see there's kind of an inflection point to these lines. So the species that have larger genomes tend to have a lower recombination rate per base pair, and the species that have smaller genomes tend to have a higher recombination rate per base pair. Also, the recombination rate is not constant throughout even the same species. You have little regions of the genome that are much more likely to experience crossing over during prophase one of meiosis. You know, they're more likely to have recombination. So Myers et al. Found that 80% of human recombination events occur in only 10 to 20% of the genome. So those are clearly hotspots
Why did they go down in the middle?
right here.
Oh, that's the centromere.
Yeah, no recombination there. That's where the sticky spot is. So the human genome can be broken up into these discrete haplotype blocks. Um, and remember haplotypes, right, are groups of loci that tend to get inherited together. And so within each block, there's not that much genetic diversity. There's not recombination, and there's very, very high linkage disequilibrium, right? But between these blocks, you have these recombination hotspots that break down linkage disequilibrium. So you have chunks of these haplotypes that get inherited together, but then each of these chunks can get separated from each other, and that chunk can end up in new combinations. All right, you made it through the end of the chapter. Go take that quiz. See you next week.